All right, welcome everyone. I see many people logging in. Thank you, thank you, thank you. So we're gonna start, okay? So welcome to Indoor Ag Science Cafe March. So this is the first actually Indoor Ag Science Cafe co-hosted by United Greenhouse Growers Association. So Melanie Yelton is here as a co-moderator. So we kind of do the dual, the moderation. Melanie may not be available every single time, but we do sort of together whenever she's available. So just wanted to go over. Oh, by the way, I'm Chieri Kubota. I'm a professor at the Ohio State University. So this cafe series is supported by now four organizations. Again, UGGA is co-organizing this cafe, and then supported by ADVANCEA group, OptimIA group, and CEA HERB group. So those additional three groups are USDA funded projects. OptimIA already ended, but still legacy, because this is a group initiated this effort, and now scope is entire controlled environment agriculture, which is in our definition, it's a from low tech to high tech. So everything, and then all crops actually. So yeah, so if you are interested to participate or spread the word to your colleagues, I really appreciate. So we provide webinar opportunities for both academic speakers and then also industry speakers. Academic speakers, you can take this opportunity as a time to share recent research findings or knowledge, or graduate students practice, public speaking, because this is not really formal presentation. This is more like an informal, although we record and archive informal presentation opportunity. So feel free to propose me or Melanie, the topics you want to present. I also welcome group presentation, like a project group, taking this advantage to showcase what they are doing, and then also what the potential impacts of what they are finding. So I really appreciate that kind of sort of creative opportunities for that. And industry speakers, introducing your company or new technologies or products to see a community. So usually, webinars, you may feel like you can't really talk about products, but I welcome actually for this one, we like to know your products. So a little bit marketing pitch is also welcome. So take this advantage because I don't think well, there might be other opportunities, but you can take 30 minutes, 40 minutes, to talk about your technology. So I really think this is a good one for you in industry sector. And then sharing experiences or lessons learned from your operations. This is particularly for growers that would be appreciated. Large growers, small growers. There may be some restriction what you can talk about, but appreciate if you can be open and share the experience so that basically the idea is, we're gonna move CA forward together. So really, knowledge exchange is something we appreciate. And then offering pre-competitive topics for collaboration, partnership. That's another great, topic if you are interested. So let me know or let Melanie know, and then we're gonna schedule your talk. So again, recordings for the presentation, and then usually we stop recording after presentation is done so that, audience can ask questions without worrying about being recorded. Um, yeah, so the three archive sites available. Some talks, sometimes, speakers do not want to be recorded or not making it a forever archive. So in that case, you may not see it, but most of the talks in the past archived. Okay, so today, March 31st, the end of March, Dr. Jason Hollick is going to talk about Vegetable Grafting 101. Basically, you learn what's going on in the vegetable grafting world and what the basic process is, and then a little bit of scientific understanding affecting vegetable grafting. This is a very important technology in greenhouse crop production, and originally, and is still very important technology in open field soil-based vegetable production. So I asked Jason, taking the advantage of having him as a lab member, to talk about this. All right. So, and then before going to Jason, we have one more talk scheduled right now. So the April 28th, Dr. Chris Curry from Iowa State University talking about herb growth in CEA, how to control the growth and development of those plants. So please look forward to that. With that, yay. So that Jason is going to talk about vegetable grafting. So I'm going to stop sharing and then have Jason.
All right. Well, hello, everyone. As Cherry said, I'm Jason. I'm a postdoc in her lab. And today I will be talking to you guys about vegetable grafting. This is one of my favorite topics. I've worked with this since undergrad, so for many years now. This has been a focus of my research. As Cherry said, I'm still in her lab. I've done a lot of work with her over the past 7 years, looking with a variety of crops, primarily though tomato and watermelon. So I work, again, my research, basically grafted plant propagation and physiology, as well as greenhouse production of high wire crops like tomato, which are generally grafted. I also do some teaching for Ohio State, focusing on sustainable vegetable production. Okay, so to start out, I just want to define a couple terms. Grafting is where we take two plants, we combine them to make one new plant. So when we talk about the scion, this is the above ground portion of the plant. This is what's going to produce our marketable product, a tomato, a watermelon. And then our rootstock is what it sounds like. This is the root system of that new plant. This was generally selected for some type of, adventitious trait that we don't have in the, in that scion. So disease resistance or vigor enhancement are reasons we might select a certain rootstock. So this picture here kind of illustrates we'd have our watermelon as a scion, and then we might have an interspecific hybrid squash rootstock as, for that root system for that new plant. We physically combine those two portions of the plant, let them grow together. So why do we graft? As you'll see throughout this presentation, there are a lot of extra steps, extra processes that go into producing an actual grafted plant, and this is also going to result in a higher cost of that grafted plant. So in order to use a grafted plant, a grower needs to have a fairly strong reason to do that, to justify that cost. So traditionally, well, I should say vegetable grafting is really not that old. We've grafted trees for a long time, but vegetable grafting has really only been, commercial since the mid 1900s. And the main reason for this has been soil-borne disease resistance. So you can, plant in infested soils, and still get a good crop. There are other reasons that we'll briefly touch on, like abiotic stress tolerance and increased resource use efficiency, but soil-borne disease resistance, primary reason, especially for open field. And then the other main reason is increased vigor and yield. So when we're talking about greenhouse crops, this is one of the main reasons that we're grafting. So soil-borne disease resistance is a very simple concept. We literally, we don't have resistance in our scion cultivar. And so we just separate the part of the plant that would be infected, the root system, and we put a resistant root system in its place. So we have pathogens in the soil, or even in a hydroponic system, you could have pathogens in that root environment, and you just put a resistant rootstock on there. to avoid any sort of crop loss. This is particularly important in soil-based systems, and even within that, in soil-based systems where there's limited rotation. So if we think about controlled environment systems that are maybe still soil-based, like high tunnels, where the same crop is being planted in the same space over and over and over again, you can get very high levels. And so grafting can be very effective and almost in some cases required in those types of settings to achieve any kind of crop. Enhanced vigor and yield, this is the other main application of grafting. This is if you're familiar with greenhouse tomato production. You're probably familiar with this. If you buy a tomato at the grocery store that was grown in a greenhouse, it's likely coming from a grafted tomato plant. So like if we think in the vein of greenhouse production of tomato, you can have instead of one plant, you can have one, or instead of, sorry, like three plants in this case, you can see this picture of this transplant here. Each one of those black arrows points to what will be a full shoot of that plant. So instead of having three different plants that are ungrafted, we can grow three stems from one grafted plant because of the vigor of that rootstock. Also, for the open field, you can have sort of a similar effect where you can actually reduce the number of plants that you have to plant, in the field because these plants will take up more space, more vigorous, can support higher fruit loads. Again, this is sort of another maybe not maybe the sole reason to graft, but could be a benefit is that typically there can be more efficient use of inputs, water, fertilizer, primarily due to the root system architecture. So larger, basically more roots and root hairs to encounter water and nutrients. Also, not really a problem in a very specific problem to certain regions, but grafting can help with salinity. So again, if you're growing in a high tunnel year after year with saline water or just with fertilizer salt accumulation, various rootstocks can help with this either through sequestration of those like sodium ions or through actually preventing damage of tissue through antioxidant production. Basically, same role for temperature tolerance. You could grow, depending on the rootstock, you could grow in, say, colder soil and still achieve water uptake and plant growth, even under what we would consider unideal conditions. So a little bit of a worldwide overview. Most of this is going to focus on U.S. Production, but worldwide, we do see grafting being adopted in a number of countries. So this map is showing any country that we have records of, some degree of commercial adoption of grafting. This may not be, a large portion of the plants used in that country, but we have some level of commercial production there for vegetables. So, tomato, watermelon, those types of things. Basically, the two regions where there's the highest rates of adoption would be in Japan and Korea, and then around the Mediterranean. So countries like Turkey, Greece, Italy, Spain, those are all countries that have very high rates of grafting. So for solanaceous crops, over 50% of those, so like tomatoes, being grafted. And then in Japan and Korea, for watermelon, it's a pretty incredible figure. Over 95% of watermelons produced in those countries are being grafted. Some of this is because in these countries, Mediterranean growing systems are, even if they're soil-based, they are very concentrated. And so again, you get buildup of soil pathogens. Another sort of, as I've mentioned, another growing system where we see a lot of grafting adoption is in greenhouse production, particularly greenhouse tomato. So in high-tech hydroponic greenhouses, we have almost 100% adoption of grafted tomatoes for that. And when we talk about the crops that are grafted, there are some other than tomato and watermelon, but these are the major grafted crops worldwide are tomato and watermelon. We have other ones like melon and cucumber. These are what we call cucurbits, along with watermelon. And then eggplant and pepper can also be grafted, just like tomato, but much less much less common, particularly in North America. Okay, so if we look just at North America, it is challenging to get obviously really good, accurate data about production and usage of grafted transplants for North America. So a lot of the data I'm presenting is from some work that was done in Cherry's lab by former grad student John Ertl. Um, he did a really good survey back in 2019 to try and get some numbers on this. We know already, though, that some of these numbers have changed. In the U.S., we have some expanded grafted capacity since these numbers were collected. So these are a little bit of older numbers, but um, the general trend is still the same. So Canada has a very high production rate of grafted plants, so of grafted transplants. A lot of these grafted transplants are being used for CEA production, either in Canada or being shipped to growing facilities in the US. So a lot of our, I'm based here in Ohio, so a lot of our Ohio greenhouses are getting, tomatoes shipped from Ontario to be grown there here in greenhouses in Ohio. So large portion of most, if not all, of the greenhouse tomatoes grown in the U.S. are actually coming from Canada. In the United States, we have definitely less grafting capacity. Something under around or maybe under 10 million plants. A lot of the tomatoes are coming from Canada. We do sort of differ from Canada and Mexico in that the majority of our transplants that we're grafting in the US are actually watermelon. So that's swapped in both Canada and Mexico. The majority is tomato, but in the US, what we do produce in terms of grafted plants is mostly watermelon. Obviously much smaller numbers, but we do have watermelon grafting. In terms of use of grafted plants in the U.S., roughly split evenly between watermelon and tomato, around 10 million estimated for both of those. Again, these are fairly rough estimates, but that's what we would assume. This is compared to some of those other countries that I mentioned, this is a pretty small fraction of our overall usage of vegetable transplants, tomato and watermelon. But again, in certain sections like greenhouse tomato, it is the majority. Mexico is hard to get data for, but we know there is very high rates of grafting, and the majority of the plants grafted in Mexico are used in Mexico. There's some, but very, very little transport of grafted plants from Mexico to the US. So again, as I've said many times, grafted tomato for greenhouse is very common in the US. We do have some adoption in the open field or for high tunnels, again, where you have soil-borne disease pressure or issues with salinity, other root zone issues. There is some growing interest from processing tomatoes. The issue there is scale, obviously, to grow, thousands of acres of processing tomato. That is a whole different scenario than growing in greenhouse in terms of plant numbers. And the main rootstocks that are used are an interspecific hybrid of domestic tomato and wild tomato. And again, this allows us to grow these multi-headed plants here. So you can see this from one little rock wool cube, we have one stem that splits into two stems to support that plant. In terms of watermelon, though, use in the US, compared to our total watermelon production, is still a very, very small fraction, maybe 2%. But this is growing, particularly in California. So we know over the past couple of years, California grafting or use of grafted watermelon has increased rapidly, and we're now somewhere over 20% of the watermelon in California is coming from grafted acreage. And one of the reasons for the higher rates of adoption in California compared to maybe other regions of the U.S. is that you can keep harvesting these grafted plants for an extended season due to climate and also availability of labor. So you can get up to 10 harvests, which is higher than what you would see in, say, Florida or Georgia. And so this allowed the fact that the rootstock can keep the plant healthy and producing for that long season is very valuable there. When we talk about U.S. production of grafted watermelon, we have two main rootstocks. So squash, again, an interspecific squash. This is if you think about like a Hubbard squash and a butternut squash. It's the basically the hybrid of those two species, and this provides again a very vigorous and disease-resistant rootstock. Squash is still majority rootstock for California. In the Southeast, which is another one of our watermelon producing regions in the US, um, it's lower rates of adoption. The reason for grafting here is primarily due to fusarium wilt. So there's race that we don't have resistance to in any of our cultivars, any of our varieties, watermelon varieties. And so grafting, you either have to graft, accept huge crop losses, or treat with some type of chemical fumigant, which may or may not be all that effective. This pathogen has a very long lifespan or very long-lived resting spores, so it's very challenging to basically outweight it by rotating crops. And in the Southeast, wild watermelon is becoming the more popular rootstock. We basically have one variety, Carolina Strongback, that's being used. And this is preferred by growers in this region because it also has resistance to nematodes. So squash also has resistance against this fusarium, but growers in this region view the wild watermelon rootstock as a benefit because of this added nematode resistance. So, a couple sort of challenges. Obviously, grafting use in the U.S. is not very high, but we know there are many benefits to it. One of the challenges is again just the straight economics for growers. Grafted plants are going to be much more expensive, and in the next several slides, we'll go through the process of grafted plant production, so you'll see why that is. But we can have much higher costs. For an open field tomato where you don't have a very long harvest season, that plant can cost up to 900% more than your an ungrafted plant. So it's rare. It would be very rare that you would be able to make enough back in that short harvest season. But something like greenhouse tomato, the price difference maybe isn't as great, and you have a much larger harvest season to receive some of that yield benefit. And so it would be more likely to be adopted in that case. But this is a pretty big barrier. Another challenge for growers is that growing grafted plants is very different than growing ungrafted plants. So it requires usually some changes in management in terms of water, nutrient management. And it can be, if growers don't have the proper support, it can be challenging for them and they may, swear off grafted plants as, too vegetative, too vigorous, too difficult to manage. And then we also have some specific concerns such as flavors, off flavors in fruit, or issues with you could have a greater overall yield and a longer harvest season, but it might push the start of your harvest back far enough that it's not economically you miss out on your best prices for your produce. So these are some of the challenges facing growers who want to use grafted plants. The price is really big, and we'll go through kind of these steps for what contributes to this. Seeds are very expensive, and you have to use a lot of them. And then the labor is another huge cost, as well as just simply all the costs associated with specialized facilities, as well as the basically you're growing twice as many plants, so you have twice as much space, twice as much heating cost, and all of that. So these are kind of the general steps. We have, pre-grafting production of seedlings, which is actually a very important step that we'll talk about. You have the actual grafting, graft healing that allows the plants to grow back together, and then, eventual shipping to the grower for production. So cultivar selection, this is actually before you even do your seeding. This is quite important. And this is important because not all scions are going to respond equally well to, say, a given rootstock. One scion might give you a much better benefit from that rootstock than another one. And so this is why growers are very often doing, doing in-house trials to determine, which of these cultivars is going to give me the best performance with which rootstock. So it's not simply, this is the best scion, this is the best rootstock. It's really that combination of the two that's important. It's very rare. You're not going to end up with a situation with modern cultivars where you're going to graft a, completely incompatible graft, unless you tried to graft like pepper and tomato or something, which doesn't work. But tomato rootstocks grow, but you may not see the maximum benefit. It's also important to consider what sort of stresses you're actually going to be dealing with in your growing environment, and then also economic considerations just relating to, which of these is actually going to give me the proper benefits. For the pre-grafting growth, this is actually a really important step. It's easy to focus a lot on the actual grafting process, but this is equally important, if not more so. And when I say the uniformity here, this is for both within your scion and your rootstock, seedlings that are going to be grafted, right? So your scions all have to be at the same stage. Your rootstocks all need to be at the same stage. And then, of course, they also have to match each other, which is a whole nother deal. But for right now, I'm just saying, your scions have to be at the same stage all at the same time so you can graft them all at the same day. Unfortunately, depending on your seed lot, this means you might have to plant more seeds than you need. So for something like seedless watermelon, this could be up to 40% more seed that you have to actually sow. And this is again for the scion, not the rootstock. 40% more scion that you sow so that you have enough ready to graft on grafting day. With, regular transplant production, there's maybe a little more flexibility. But really, with grafted plants, we need to be able to graft as many as possible on the same day. It's really hard if you get to the point where you're trying to split orders and hold some plants back to be grafted at a later date. So you really need to have uniformity. In addition to that, the scion and rootstock have to match each other. So typically, we think about like stem diameter and overall size of the plants. And in order to achieve this, generally, this means you have to seed at different dates. So this is the easiest way to control this is, for every seed lot, for every cultivar you have, how many days it takes under your conditions to reach graftable size, and then you can stagger your planting accordingly. So in this picture here, you can see that our seedless watermelon is ready to graft in 21 days, whereas our squash rootstock is ready to graft in as little as 7 days. So we plant them ahead of time to account for that. And again, testing for both uniformity and time to grafting is very important. So even for specific seed lots, there can be big differences. Obviously, that you can also do manipulation of the environment in order to try and alter your plant growth. If you need to speed up growth, slow down development, if you need to stretch or prevent stretching of stems. So all of this can be somewhat controlled in your environment. One thing I'll mention is for field-based production, it's really important to have a proper length rootstock so that you don't have contact of the scion with the soil system or with the soil where you might have pathogens that can infect it. So there are many different techniques you can take, obviously, depending on whether you're growing in greenhouse or growing in a completely controlled indoor setting. You may have different options for control, but things like photoperiod, light intensity, even the spectrum of the lights, these can all have a large role on altering plant growth. Or even just, if you're in a greenhouse, moving plants from a cool side of the greenhouse to the warm side of the greenhouse. Just again, trying to make sure they're all matched up. Now, moving on to the actual grafting steps, we have two main methods of grafting that we use. One for cucurbits and then one for solanaceae. So like our tomato or eggplant would use a different type of grafting. So for cucurbits, we call it single cotyledon grafting. This is because we leave a cotyledon on the rootstock. So this is actually in the graphic. It's the second half of that, the lower half of that, and you can see we leave one rootstock cotyledon on the plant. Splice grafting is more simple. It's literally just an angled cut on, our scion, on our rootstock, and those sections get stuck back together. Regardless of the type of grafting that you're doing, you're generally going to use some type of, silicone or plastic clip to actually hold that cut together until the plants can grow and rejoin each other. Okay, so I am going to go quickly through the steps for grafting. I know for some of you, if you're not, doing the propagation, this may not be the most interesting, but I think it is important to see it and understand it, particularly the single cotyledon grafting, because it is a little more challenging. Um, in this case, I'm going to be using watermelon and squash as an example. Squash is interesting in that we actually tend to graft this without the root system. So we actually cut the rootstock, at the base of the substrate. And then while the graft is healing, it's also growing new roots. But for other rootstocks, that wouldn't be the case. We would still have, a root ball attached to the rootstock. So basically, we call it single cotyledon because we save one cotyledon on the rootstock. We get rid of the apical meristem and the other cotyledon. And there is, as you'll see in a couple later slides, there's a little, see if I can, there's a little tiny cotyledonary meristem that's basically at the base of this cotyledon, and that has to be removed. You can't really even see it in this picture, but where my laser pointer is, that's where it would be. And that's what you have to remove to ensure that there's no rootstock regrowth. For the scion, it's much simpler. You just make an angled cut below the cotyledons, and then you attach those with a clip. And then if you're using squash rootstock, this gets rooted into the substrate. So this is a video, hopefully. Okay, I think the volume's off. If it's not, it's going to play some obnoxious music. So hopefully that doesn't happen, but we'll see. It's also, the video is a little slow, but I do think it helps to see the process. So again, for a squash rootstock, we graft it as an unrooted cutting, essentially. So this is removing the apical meristem and the other cotyledon while saving the other cotyledon behind. This is the cotyledonary meristem. This is the hardest part of this because you have to remove this completely without damaging, your other cotyledon or any of the stem. And then it's just about lengthening the cut if you need to, so that you have good surface area contact between your scion and rootstock. So then for the scion, it's a lot simpler. It's just the angled cut. So then those just get stuck together with the clip. And then it just gets inserted into the substrate like that. So this is what happens if you don't fully remove the cotyledon meristem. So you can see this little red circle that is around the tiny little cotyledon meristem. If you don't remove that, you end up with here where you see those divided leaves in this in this figure. Those are the watermelon leaves we want that we grafted on, but then we also see these larger, lighter green leaves coming from the base of the cotyledon. This is basically outgrowth from that cotyledon meristem. So this is why it's really important to, you have to have well-trained grafters, because if you sell a farmer a bunch of grafted watermelon that produces, pumpkins in the field, they're not going to be happy with that. Splice grafting is a much simpler process. This is again what we use for tomato, eggplant, those types of crops. You can see in this picture here, um, right here around this pin clip, you can actually see there's this nice scar that shows perfectly that angle that we'd be looking for both of those. So the steps are very simple. Angled cut. For the rootstock, you always cut below the cotyledons so that you don't have, there's no risk then for rootstock regrowth in this case, as opposed to the single cotyledon grafting. Grafting clips get placed on there. Same cut made on the scion. Where on the scion can change a little bit. This depends on the specific end use. So typically, at least for greenhouse production, these plants are going to be pinched to create multiple shoots. And so if you're going to pinch at the cotyledons, you obviously need to leave the scion cotyledons. But if you're going to pinch after the second true leaf, you actually might want to graft above the cotyledon so your plant's not as tall. And again, you don't need any shoots to grow from the cotyledon, so you can graft above the cotyledons on the scion for that case. And then it's just a matter of inserting those two scion and rootstock together and making sure that every time when I teach students this, they struggle, but it's common sense. The clip just has to hold the scion rootstock together. So that's all this plastic clip is doing, is maintaining a little bit of pressure on that graft union. And then if needed, stabilize with a stick. So particularly the single cotyledon grafting is quite challenging. Good watermelon grafters can do over 150 plants an hour. I have seen as high as 280, but that is, very, 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 very rare that's possible. Generally much lower than that. Tomato grafters typically would be able to graft over 200 plants an hour. And so this is basically a skill that requires a lot of training to get good at. Everyone can graft a tomato, or a watermelon, but to be able to graft them well and graft them quickly requires a lot of practice. And it's not going to be a permanent job that's year round. So with that, there has been a lot of interest in grafting machines. There have been grafting machines produced for the past 30 years, but still fairly limited commercial adoption of those. It's still mostly hand grafting. Another challenge is it's fairly easy to mechanize the tomato grafting, but for cucurbits, it's a little more challenging because of the precise nature of that. But we do for tomato, there are lower cost, I should say, semi-automated machines that can graft, 400 plants an hour. And then other larger scale machines that are, much more expensive that would be able to graft basically 10 times more than that in terms of production. So this is an example. This is actually one that we have here at Ohio State. Just a small model developed by Kusakabe in Japan. And it just allows us to, you still have to have someone to run the machine, but they don't necessarily have to have the same training or experience, or they could also do other jobs. They don't need to be specialized in grafting. So this video that's about to play is pretty short. So it's going to be over on the right-hand side pane. So just watch because it's kind of zoomed out. But we just put the plants in the machine and it is going to make the graft for us. And obviously, if we were doing a whole bunch rather than just taking a video, you'd be feeding the next set of scion and rootstock into the machine while it's spitting out the other graft. One quick note, we always say that grafting is really good for disease resistance. but it also comes with the risk of introducing disease. If you have one plant that has a virus in it or some type of bacteria, and then you go and cut that with a blade and then cut other plants with that same blade, there is the potential to spread diseases fairly quickly. So it's good to swap out blades or sanitize, and obviously sanitize your work surfaces as frequently as possible is always better. So most places will just use disposable razors for that purpose. Now, the grafting process, speed is important from efficiency, but also the fact that we have these plants are actively growing, and so you need to then be able to get them into high humidity conditions as soon as possible so that they don't desiccate, so that they don't wilt. We basically want that high humidity, stop the transpiration until we have reconnection of the xylem and phloem, until we have water flow from the rootstock to the scion. So this is the process we call healing. And this can be as quick as 5 days, generally longer, 5 to 10 days. Process depends a lot on the plant age and obviously the type of plant. But you don't need a lot of light in this period. The main focus is maintaining high humidity. So these are two examples from like greenhouse focused healing, so benchtop healing, where you would have tents that maintain high humidity around the seedlings, and you can see, we have some shading going on so that there's not, very high light intensity. Another very common option, particularly for plants going into CEA, but also for plants going into the field, would be completely indoor controlled healing conditions. So in this case, you would have air conditioning, you would have and some method to maintain high humidity. So typically like a dry fog system. And then again, the light intensity doesn't need to be very high. So this could be lights in different racks or lights on the side of the room. These are both examples of options for that. So you can this is some examples again in sort of cartoon illustration where you could try and maintain the high humidity in the whole room. But then you can also actually do it basically on an individual rack basis. So instead of trying to maintain the whole room at a high humidity, you can wrap those racks in plastic or some other barrier to maintain high humidity inside. And then that can also help with targeted reduction of humidity, because you can then put holes in that plastic to decrease the humidity as you need to. As that plant gets vascular reconnection, you eventually need to introduce it back to ambient greenhouse humidity. So once all of that's done, once the plant has been healed, then there may be some additional steps that need to go on, or some additional growth before they get shipped, particularly if you think of like a greenhouse tomato that's shipped as a fairly large transplant. So in this case, there might be some pinching, like I mentioned earlier, or there might be some other sorting, quality check, size grading, that type of stuff that goes on after grafting. But then at that point, plants get shipped. One difference compared to say obviously even standard transplants that aren't grafted are going to be shipped. But generally that can be closer, aren't traveling as far. And in some cases for grafted plants, they don't travel very far. You can see these blue squares in Ontario. Those would be shipping tomatoes to, as close as Ohio. So that's not a very long trip. But if we think about this green square down here in North Carolina, that's producing grafted watermelon. And that's being shipped all the way to California. So, a several day journey, on a truck out to California. So just another stress that these plants have to deal with, and an extra shipping cost. So a few things as I'm wrapping up, I just want to mention some things we're seeing is some interest areas in. I have new crops in quotation marks here because they're not really new crops. They are somewhat grafted. We know how to graft them. It's not a brand new thing, but we're seeing emerging interest in these. So for greenhouse pepper, this is typically not grown as a grafted plant, but due to some fusarium pressure and other root issues, this is being seen. There is some growing interest in grafting pepper. Greenhouse cucumber is another one where it's typically not grafted because the production in the greenhouse is not a very long harvest cycle. So again, you may not see enough benefit in that short cycle to justify the extra cost. Um, but again, due to disease issues, and potential for season extension, there is some growing interest in the U.S. and Mexico particularly Mexico, for growing grafted cucumber. Also with some new rootstocks that are true cucumber rootstocks that don't necessarily have the same um, concern over altered flavor of the fruit. And then, as I mentioned before, there is some interest for open field processing tomato growers to start using grafted plants. The challenge there again is scale. Even with reduced planting densities, you still need huge quantity of plants in a very short period of time that we just currently can't can't meet without increased use of mechanized grafting. Another thing, I just wanted to throw this slide in here because I find it fun to just to see all the kinds of things you can do with grafted plants. I think it's really incredible from a teaching standpoint and just from getting the public interested and involved with horticulture and agriculture. You can do some pretty cool stuff. You may have heard of ketchup and fries. So we have like a potato rootstock with the tomato scion. You can graft different plants together. 5 different tomatoes on one plant. So there's lots of cool stuff you can do with grafting, but these aren't necessarily commercial applications or anything like that, but just fun and cool to mention. So to wrap up, grafting can be a very valuable tool under the right conditions. I'm not going to say that grafting is the perfect solution for everything. You have to have an economic justification for using grafted plants. So that's one of the challenges for growers who want to use grafted plants, is particularly for field-based growers, is that the plant cost is still very high. But we are seeing continued interest and adoption of grafting in all sectors in the US. But again, the challenges: high cost of plants, challenges for growers to get used to growing grafted plants, and then also, there can be some other more specific challenges like harvest being pushed back a little bit, or specific fruit quality concerns. So, but overall, again, it's just going to come down to the specific situation, whether it makes sense or not for someone to be using grafted plants. And so with that, that's all I have. If you have questions, I'll take them now, or you can also reach out to me over email if you have any specific questions.
Okay, great. Thank you, Jason. Very informative. Lots of information.